but in regards to our perio class. So, basically, what's the problem with immunity and inflammation? There are 10 to 50 trillion cells and 100 trillion to 1 quadrillion bacteria. And our hands and body can have lots of microorganisms and bacteria. Bacteria outnumber human cells 10 to 1 in each person's body. So, because of all this bacteria, we have what's called the immune response, or immunity. The prime purpose of the immune system is to defend the life of the individual by identifying foreign substances in the body and developing a defense. How much of the immune system lies in the gut, or the gut health? 70%. 70% of the immune system is in the gut health. The host response is the way an individual's body responds to the infection. So the host response is how the body responds to the infection. So, and what affects the host response can be many factors such as stress, diet, age, etc. Malfunction. So, when you have immunity malfunction, AIDS is an example of a loss of immune function, and it can be deadly. You have HIV virus, which disables a specific group of immune cells, and then HIV positive individuals can develop infections from microorganisms because they have poor immune systems. Another malfunction of the immune system is overzealous repair. Overly intense response harms the body while trying to protect itself. This is when your body is creating a lot of inflammation towards bacteria, but it's working too hard and going too crazy, such as in periodontal disease. And also Hashimoto's, lupus, rheumatoid arthritis, anything like that. Chronic periodontitis is an example of an overzealous immune response. Some key points. Inflammation causes most of the damage in periodontal disease. Inflammation is low grade but continuous and a constant onslaught to the periodontium. So we talked about how you can talk to your patients about how a diabetic person with a foot wound is the same type of wound in a periodontal infection. Both are biofilm infections requiring debridement, or removing the biofilm. What is the likelihood of infection without care or treatment? Very high. If you have an infection going on and you don't treat it, your likelihood of getting it worse is very high. So, in the immune system, what are the players? These are the cellular defenders. You have phagocytes, which are neutrophils or PMNs, and then macrophages, and then you have lymphocytes, which are the B cells and the T cells. You also have immunoglobulins, also known as antigens, and then you have the complement system. And just a side note, what are the defenders that are the only ones that release most cytokines? Those are PMNs, neutrophils, macrophages, and T cells. So here's an example. So what's the very first cell to arrive? The mast cell. The mast cell is the very first cell to arrive. And the mast cell has to be there first only in the situation of trauma or infection. So, generally, the first cells that arrive are neutrophils, right? We know that. Neutrophils or PMNs. But in a situation with trauma or infection, the mast cells are the first to arrive. And this is just a chart showing you, again, where each category lies. So lymphocytes are B and T cells. Phagocytes are neutrophils and mononuclear phagocytes. And then helper cells are mast cells, basophils, etc. Cell differentiation. So, in the blood, you have stem cells that differentiate into two cells. So on the chart, you have a blood stem cell, and it can turn into one of two things: a myeloid stem cell or a lymphoid stem cell. If you go down the myeloid route, the myeloid cell can turn into three other things: It can turn into red blood cells, platelets, or myoblasts, which then turn into granulocytes, basophils, neutrophils, eosinophils, etc. Okay, so neutrophils, those can turn in from myeloblasts. Now, going back to the blood stem cell at the top of the chart, you can have a blood stem cell and it can turn into a lymphoid stem cell, and that can turn into a lymphoid blast, which then can turn into three things: a B lymphocyte, which then turns into a plasma cell, or a T lymphocyte, or a natural killer cell. Okay? So, also important to remember, where do B lymphocytes mature? In the bone. B equals bone. Where do T lymphocytes mature? In the thymus. T for thymus. Okay, leukocytes. Leukocytes act like independent single-cell organisms. They can move through tissues capturing microorganisms on their own. In periodontal disease, the leukocytes are PMNs or neutrophils, and monocytes or macrophages. Each leukocyte acts as their own unit, independent, but they can also communicate with each other. So, for example, we talked about a cake, right, in class. Think of a cake to be in your connective tissue, and there's a fly on it. And you go and swat the fly, and your fingers take off some of the frosting of the cake. What is left without that frosting? Just the cake, right? So, did the cake get destroyed by swatting the fly? Yes. So, for example, in the example of connective tissue, leukocytes are going to destroy tissue in order to get to the area of need, and that's important to remember. Therefore, they're considered destroying cells. Okay, PMNs, polymorphonuclear leukocytes. These are also neutrophils. PMNs, neutrophils, same thing. Now we talked about, generally, these are the rapid first responders. They capture and destroy bacteria invaders, and they're short-lived. Why are they short-lived? Because remember, neutrophils, once they phagocytize or kill, they die, just like a bee sting, a bee dying with a bee sting. Okay, they are attracted to bacteria by chemotaxis. They contain lysosomes, which are strong bactericidal and digestive enzymes that help kill and digest. Periodontal pathogens are most effectively destroyed by neutrophils. So, in periodontal disease, what are the cells that help destroy most pathogens? Neutrophils, or PMNs. Some medications can cause neutropenia, which is when you have diminished numbers of PMNs, and that can increase the risk of infection. And if you have acute infections going on, you're going to have high numbers of neutrophils. Okay, so now you have monocytes and macrophages. Monocytes are in the bloodstream, and macrophages are in the tissue. So remember, it's like our chart. Monocytes, and then once it's out of the blood cell, it's a macrophage. Sorry, once it's in the bloodstream, it's a monocyte, and then once it's out of the bloodstream into the tissue, it's a macrophage. Macrophages are slower to get to the infection. That's why they're second and they don't come first. Who comes first? Neutrophils. They also surround and destroy bacteria, but these cells live longer because they don't die after phagocytosis. And they help present the antigen to T cells. Lymphocytes. Small leukocytes that help defend the body. What are the two types of lymphocytes? B cells and T cells. B lymphocytes, B cells. These defend against bacteria and viruses. They differentiate into two types of B cells, plasma B cells and memory B cells. Once activated, they make millions of antibodies that pour into the bloodstream. So, what cell arrives first, the plasma B or the memory B? The plasma B arrives first, but the memory B remembers the antigen, and therefore they make the antibodies as a weapon. So, again, two types of B lymphocytes, plasma B cells and memory B cells. We talked about antibodies being in the shape of a Y. So antibodies, Y-shaped proteins. They come from plasma cells, B plasma cells. They're also known as immunoglobulins. So antibodies, immunoglobulins, same thing. The stem of the Y binds to the outside of the B cell, and the arms of the Y bind to the microorganism, kind of hugging it to inside of itself to kill it. Antibodies come from the plasma B cell. Antibody defense mechanisms. Antibodies neutralize bacteria. They coat the bacteria, or like we talked in class, they spray paint the bacteria so that we know for sure they stand out for phagocytosis, and they activate the complement system. Immunoglobulins. Antibodies are also known as immunoglobulins. 5 major classes of immunoglobulins. So you have IgG, IgA, IgM, IgE, and IgD. And remember, where do antibodies come from? Antibodies come from plasma cells, which come from the B lymphocytes. So IgE, what's that one? IgE is the first antibody you're born with. It makes up 3/4 of all of our antibodies. The other 4 take up 16 years to develop. So IgA, the A team, has to do with saliva, tears, mucous membranes, and vaginal secretions. It's kind of the body's way of releasing bacteria through those routes. IgM has to do with bacteremia. IgM bacteremia. It's the first antibody if there's any bacteremia or blood infection. It's most prevalent with autoimmune diseases. IgE, remember E equals parasite. IgE fights parasites. It's also found in allergic reactions. And then IgD equals dispatch and diminish. IgD can either stimulate or depress the antibody-producing cells, and it's a regulatory immunoglobulin. So IgG, what we're born with, 3/4 of our antibodies are made of this. IgA is the A team, saliva, tears, mucous membranes. IgM has to do with bacteremia or blood infections. IgE, E equals parasites. They destroy parasites. And then IgD, dispatch and diminish, stimulate or depress the antibody-producing cells. Okay, T lymphocytes, T cells. T lymphocytes intensify response of macrophages and B lymphocytes. They produce cytokines, like interleukins, which further stimulate the immune response. Cytotoxic cells are T killers, helper cells are T helpers, and regulatory cells are T regulators, or suppressors. What other 2 cells also produce cytokines? We know T cells do, but neutrophils and macrophages also do. Cytokines. Cytokine is a general name for any protein that is secreted by cells, and it affects the behavior of another cell. The complement system. The complement system is a series of proteins circulating in the blood, complementing the work of antibodies. It's called into action by immunoglobulins. It's activated when pathogens attempt to avoid immune cells. The complement system facilitates phagocytosis of bacteria. It kills bacteria directly by forming pores in bacterial cell membranes. So what are the functions of the complement system? Well, we want to destroy pathogens directly, also known as a MAC attack, putting holes in the cell membrane, lysis. It uses opsonization of pathogens, which is the most important action. Opsonization is where they coat the surface of the pathogen for swifter recognition. So basically, you're spray painting the pathogen so it stands out bright. That's opsonization, which again, opsonization is the most important action of the complement system. The complement system also recruits phagocytes. It calls in for support, and it's an immune clearance. It acts as a housekeeper for the body by removing immune complexes. Okay. Functions of the complement system. This is just a chart talking about what we just talked about. It can lysis or kill cells. Opsonization, which is the most important one, which increases phagocytosis by spray painting the bacteria or coating it. It is an activation of inflammatory response, and it has a clearance of immune complexes. Also, let's note, opsonization can be called many things. It can be called extravasation or emigration or transendothelial migration. Okay. And what cell creates permeability in the vessel so that the other complement receptors can escape the blood vessel? Mast cells. Okay, and then leukocytes. What do they do? Leukocytes travel through the bloodstream to get to an infection. They squeeze through the endothelium blood vessel lining. They are involved in extravasation, which is when the leukocytes make their way past endothelial tissue to connective tissue. Process is known as transendothelial migration. Chemotaxis. It's a law of attraction for leukocytes. Biochemical compounds are released by bacteria attracting leukocytes. So yeah. Phagocytosis, we know this is when the leukocytes engulf and digest microorganisms. How does phagocytosis work? Well, the bacterium becomes attached to the membrane in vaginations called pseudopodia. Bacterium is ingested, forming a phagosome. Phagosomes fuse with lysosomes. Lysosome enzymes digest captured material, and digestion products are released from the cell. Phagocytosis, tissue destruction. Once lysosome enzymes are released, the surrounding tissue is destroyed. Because just like in this picture we talked about, here's the bacteria getting engulfed, phagosome fuses with lysosome, and then they pop out of the cell again, and this tissue is becoming destroyed from that. Alright? Cool. Basics of inflammation.